Welcome to Dr. Whiteboard Talks once again. Today's topic is functional anatomy and physiology of nervous system. So we know that nervous system is comprised of multiple billions of cells, neurons, and the supporting cells, which is connected, interconnected, very sophisticated manner, where it forms the whole brain and the spinal cord, plus the peripheral nerves and central nervous system. So right now, the nervous system in brain, how it is formed, is what our topic today. So brain means all the central nervous system, what I'm talking about. So if you see the observe, and if you see to observe this diagram, you have uh, you have to observe here we have neurons. What we say neurons is like the nerves forming, the nerve cell forming the brain and the spinal cord is neurons, and they have the supporting cells. So what are the supporting cells? So functional anatomy means we'll be dealing with which are the basic uh, basic elements of the brain which is formed, and what are the cells supporting the nervous system. So if you observe, we have neuron, plus we have the other supporting cells like astrocytes, we have oligodendrocytes, and we have ependymal cells. So we have ependymal cells, which are the cells which are lining the cerebrospinal fluid cavities, which means the ventricles, and where the CSF is produced. So if you see, this is the CSF which is produced by the ependymal cells and the choroid plexus from the ventricles. So we have astrocytes, oligodendrocytes, ependymal cells, and we have microglial cells, which are the small supporting cells inside the network. So this is the schematic diagram you see about the supporting cells and the neurons of the brain. And in the other area, in the other side, you have the spinal cord and the peripheral nerve coming out of the spinal cord. So we have the peripheral nerve here, this one. With the peripheral nerve we have myelin sheath. So there is if you observe, there is myelin sheath, and we have the node of Ranvier in between, and these are the nodes of Ranvier. And so coming to the topic, we know the cells of the central nervous system or the nervous system is comprising of millions and billions of specialized cells, forming a spectacular network of connections, which means the synapses mostly, plus other connections, which are the supporting cells. So in addition to the neurons, this is what I told, addition to the neurons, we have three types of glial cells. These glial cells are the supporting cells. So what are the cells? First one is astrocytes. So astrocytes form the structural framework for the neurons and control their biochemical environment. So the function of astrocytes is that they are the one which form the structural framework of neurons, control the biochemical environment, and their food process forms the blood brain barrier. So they have a food process which forms the blood brain barrier. So astrocytes, if you come to this image, you can observe astrocytes are those cells which are like these kind of cells. If you observe, they have a food process. These food processes are those which form the blood brain barrier. So this is the food processes of food processes of such astrocytes which form the blood brain barrier. So these are the astrocytes surrounding the brain capillaries and the neurons, forming the blood brain barrier. The blood brain barrier are those which are the tight junctions in the brain, which does not allow the passage of specific molecules into the brain, whereas it allows some other certain molecules can to enter. So there is a selective uh, permeability in the tight junction, that is the blood brain barrier, and it's a protective mechanism. Once we'll be dealing it later. So this is the first case, and the second thing we have is oligodendrocytes. Oligodendrocytes are those cells which are responsible for the formation and maintenance of myelin sheath. So myelin sheath is formed and maintained by oligodendrocytes. So these are the ones which surrounds the axons and they maintain the speed and the consistency of action potential. So action potential once generated, it should be propagated, and the propagation of the action potential, the speed of which is maintained by oligodendrocytes. Peripheral nerves have axons invested in myelin sheath. So peripheral nerves are the axons which are invested in the myelin sheath made of oligodendrocytes, and these are called as Schwann cells. So you know that Schwann cells are those cells which are covered in the peripheral nerves, which are covered, which is made up of again the oligodendrocytes, that is Schwann cells. So we have the other two cells, which is one is microglial cells. Microglial cells are the monocytes or macrophages, which have a role in fighting infection and removing damaged cells. And we have ependymal cells. We have ependymal cells are the cerebral ventricles, which are lined, and this ventricles from where the choroid plexus is, and the CSF is produced. So basically, ependymal cells are those the lining cells of uh, ventricles. So to sum up, we have four cells around: astrocytes, oligodendrocytes, microglial cells, and ependymal cells. And the structural picture is like you have ependymal cells, you have astrocytes, you have the food processes which causing astrocytes. The food processes cause the blood-brain barrier. It was a small topic based just for the structural framework of the cells in the central nervous system and the supporting cells. Thank you.